Hey everybody, how's it going? I hope that you had an awesome spring break. Today we're going to be talking about protein synthesis, and more specifically, we're going to be talking about the first step of protein synthesis, which is called transcription. I'm going to go back to our last lesson though, just quickly here. We talked about RNA. So remember, RNA is a little bit different than DNA. We talked about the similarities and differences. So for the RNA, one of the most important jobs that it has is to create proteins. So protein synthesis is just a fancy way of saying like it creates proteins. So we know that we have our DNA in the nucleus of the cell. So the DNA actually codes for our proteins. And proteins are important because they make up everything on Earth. So, like our proteins, they create our tissues and our organs, or our tissues which create or which make up our organs. And of course, our organs are vital to everyday life for everybody. So yeah, this is definitely a really important process, and we're going to see how the RNA works into that process. So there are two parts to protein synthesis. The one that we're going to talk about today is transcription, which is the first half of the process. And that's going to be how the RNA is going to be coded to make the proteins in the first place. So just looking at transcription, we know that the DNA is stuck in the nucleus of the cell. So how do we get that genetic code out? Well, we use RNA for that. So just how we did DNA replication before, there's also going to be RNA replication. And as we know, the RNA is able to leave the cell and go to the ribosomes, where they can make amino acids, which make up proteins. So the first step is very similar to DNA replication, where the DNA is unzipped. So here we can see these strands on the left, they're all connected in the middle by hydrogen bonds. But these strands here in the middle, they are going to be unzipped or broken up, basically, those bonds. Then there is an enzyme called RNA polymerase, which grabs the RNA base pairs, and it's going to copy the DNA strand. And what that does, sorry, move this out of the way here. There we go. So it creates a strand of mRNA. So if you to the last lesson, mRNA, the M stands for messenger. So what it's doing, and it's going to send that message out of the nucleus to the ribosome. So you can see here, this is the strand red band, and you can see how the bases match. Remember, C goes with G, G goes with C. If there's A, it's an A on the mRNA, but if there's an A, it doesn't create a T, because for RNA there is no T, but there is a U for uracil. And then after it makes the copy, it just leaves the nucleus, and it just travels out. It just travels out through the nucleus with that copy of the message. And remember as well that RNA is just a single strand compared to the DNA here, which is double-stranded. I'm just going to move myself over here. Yeah, so that's going to be it for today. I'm going to post another video just showing another example of the way that the mRNA gets its copy, basically. But yeah, just wanted to keep this video a bit short today, and I hope you all have a great day, and I will see you again soon. Bye.